This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on March 11, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today we are coming to you from Austin, Texas. We're at the University of Texas, and right here next to me, from Austin, Rich Condit.
Hi, everybody. I'm just looking up the weather. It's, by the way, do you do the weather on TWiV? I did the weather yesterday on TWiV. Was that unusual?
Sometimes. It's really just a thing on TWiV for the most part.
It's 64 degrees outside. It's absolutely gorgeous. This is the best time of the year. The bluebonnets are up. Man, driving up on Mopac is just lousy with bluebonnets and whatever those orange things are and stuff.
What is Mopac?
It's a road.
That's what we did yesterday.
It was a different road. Mopac is special. They got wildflowers galore on Mopac.
We went to Johnson City yesterday.
Yeah. At any rate.
Went to the Science Mill and recorded a TWiV 100 there. It was really, have you been to the Science Mill? It's very cool.
Yeah.
I mean, I was impressed, actually.
Yeah, Science Mill is great. And it was packed yesterday. You know, what was yesterday? Sunday?
Yeah.
First weekend of spring break. It was just crawling with kids, you know, all having a good time. It was really great.
We have a guest for you today from the university. He's here for his fourth TWiV. You count them, right? Jason McLellan, welcome back.
Thank you very much. Excited to be here, part of the Four Timers Club.
Is that more than Kartik?
I think it's close.
Yeah, he always wanted to beat Kartik Chandran in the number of TWiVs.
Do you get a jacket at 5 appearances, like on Saturday Night Live?
Yeah, we could do that. Hey, it's a good idea. A Microbe TV jacket. You want a shiny jacket, right?
Yeah.
Okay.
Or
Sequin jacket.
Maybe not that shiny.
There is such a thing? There's a Microbe TV jacket?
No, you just gave me an idea.
Okay.
I think it's a great idea.
Because I don't have one.
For the 5 timers, yeah, it's a good motivation.
Okay.
We'll get you one because you have 1,000+ episodes, right?
Not quite 1,000 because I've missed a few, but if you want to say it's 1,000, I'm in.
We have had Jason on TWiV 500, which we were here for. His room was full for that, and I recognize many of you.
That was pre-pandemic?
Oh yeah, pre, yeah.
That was TWiV. That was my birthday.
Yeah, it was. Yeah.
That was I turned 70. Wow. So that was
so
5.5 years ago.
You're really old.
I am really old. So that was June 29th, 2019? Yeah, probably.
So we had you and Jan also. You're a guest. Who else was a guest? We had the lady, the pro-vaccine lady, Jenny.
Jenny, right.
Remember?
Sue.
Jenny Sue. Yeah. All right, so that was 500. Then you were on 578. That was the monoclonal antibody company. Prometheus project.
Oh, yes. Yeah, that was our U19 consortium.
Yeah. And that was still pre-pandemic. It was up because it was a big retreat up in upstate New York somewhere. Yeah.
That must have been fall of 2019 then?
Maybe. But certainly pre.
Okay.
Certainly pre. And then 714 during the pandemic.
I think that was a remote appearance.
Yes, it was a Zoom thing, yeah.
It's cool how we now mark time with the pandemic.
Yeah.
Especially relevant in the current context.
Yeah, I think that's right. I was on for like all things Spike.
The Shape of Spike with Jason McLellan.
Yeah.
Yes. Yeah. It's not a dimer, folks. But today I want to talk about a variety of different things. But first, if you enjoy what we do on these programs, please support us. We depend on your support. We don't do ads. We don't monetize. Our philosophy is our listeners should support us. We try and provide a free education and in turn support us so we can do these things. So you go to microbe.tv/contribute. There are a variety of ways that you can do that. A cup of coffee a month, 2 cups of coffee a month. How's that?
I enjoy what we do, and I'm not giving you anything.
I know.
Get it?
Well, you give us the talent.
I'll give you a ride.
That's good. All right, and now you gave us a ride yesterday. That's right. It's an hour each way. It was a big deal. Yeah, thank you. I know we covered your career in every episode, but Peter Palese used to tell me it never hurts to repeat things, especially to medical students. He used to tell me. So repeat your career.
Starting from when?
So you're from Connecticut? Is that
No.
where are you from?
See, Vincent doesn't
Yeah, I
even
don't
remember.
remember.
Just outside Detroit, one of the suburbs in Michigan.
Michigan. Okay.
Yeah, I went to university at Wayne State University in Detroit. It's one of the big commuter schools. Majored in chemistry with an emphasis in biochemistry. Did a couple of years of organic synthesis in the lab, and then a final year of biochemistry, purifying proteins, and really loved that. Went to Johns Hopkins School of Medicine in Baltimore for my PhD. Worked with Dan Leahy, who's in the audience.
Yay. Good job.
So I fell in love with structural biology, learned X-ray crystallography, worked on the hedgehog signal transduction pathway. And then for my postdoc, went to the National Institutes of Health, so just a bit south of Hopkins. And there I was in the lab of Peter Kwong, who was pioneering approaches for structure-based vaccine design. It's applied to HIV-1. And then I started collaborating with Dr. Barney Graham, who's an expert on respiratory syncytial virus, and began working on RSV as well. 2013, started my independent lab at the Geisel School of Medicine up at Dartmouth College. That's the New Hampshire area. I was there until early 2018, and then moved down here.
Geisel is a Dr.
That's
Seuss,
right. Yeah. So Dr. Seuss's estate.
Theodore
gave
Geisel,
a
right?
Substantial. One of the most famous alums.
So what drew you to science initially?
That's a good question.
The money,
It's definitely not the money.
the fame.
Yeah, maybe. I don't know. When I went to university, I was pre-med. It just seemed like being a doctor. I didn't know any scientist or anything, kind of a blue-collar environment growing up. And then I just loved organic chemistry and decided,
that's weird.
No, it was class averages were like 60%, and I had like 120, and I was like, okay.
They hated you, right?
Yeah, I was really breaking the curve. So then the professor teaching it asked me to like work in his lab, and so I was like, man, I just fell in love with the lifestyle of being in the lab, working with grad students, postdocs, staying late, being on the weekends.
But you entered college pre-med.
Yeah.
Was there a science motivation there, or you just thought, I have a brain, I ought to use it for something?
I think, I don't know, I was valedictorian of my high school, right? I think smart kids, the only things I knew were like
You ought to be a doctor.
engineer, doctor.
Yeah, one of the things I figured out at some point along the way is that you shouldn't be a doctor unless you really want to take care of patients.
Yeah.
Okay.
Yeah. So I was in a
And that
hospital.
wasn't me.
Yeah, I was in a hospital for a little bit, and just realized that wasn't for me. Just the smells were overwhelming.
So you don't have any regrets not being a physician?
No, I think it's all worked out well.
Good. Yeah. In fact, you're getting some award tomorrow. Tell us about that.
Yeah, it's from Research America, which is an organization that advocates for science funding. And this is the Building the Foundation Award. So I think I'm the fourth winner of this award.
It's great. I like that organization. Yeah.
Yeah.
Yeah, I think they do a lot, just trying to raise awareness, raise funding, and help people understand the benefits of science, basic science research.
So their CEO, whose name I'm forgetting. Anybody remember who she is? I can't remember. Anyway, she has this great saying, which I use all the time. When someone asks who you work for, tell them, I work for you. So if you're in an elevator and some lay person says, what do you do? Who do you work for? You don't say, I work on Drosophila, because they'll leave. You say, I work for you, and then because you're taxpayers, right? And you do work for them. And that's a little lead in to talk about your work, right?
Yeah, that's nice.
I use it at the end of my talks on SciComm, which is, if you don't want to do anything else, at least do that when people ask you what you're doing. And then if they ask you more details, that's great. All right. I want to talk about, also, because repetition is good, I want to talk about two prolines. And you know what I'm talking about. And everyone else will learn eventually, because then we'll use that as a jumping point. And so where did that come from, the stabilization of spike proteins? Where did it come from? and tell us how it works and so forth.
Okay, yeah. So my lab, back when we were at the Geisel School of Medicine, designed 2 proline substitutions in the S2 subunit of the spike protein, and showed that for many of the beta coronaviruses, essentially all of them, it boosts expression of the spike protein when we produce it in the lab and keeps it more stable. So that was a collaboration with Barney Graham as well as Andrew Ward's group out at Scripps Research Institute. And so we were working on coronaviruses well before the pandemic. So I think we started in 2013, right after the Middle East respiratory syndrome coronavirus had emerged in Saudi Arabia and the surrounding region. We felt like we really need to start learning how to make coronavirus vaccine antigens. Because we had SARS in 2002, MERS in 2012. We felt like there'd be another one maybe 10 years later. And so Andrew's lab was able to get the structure of the HKU1 spike protein, which is one of the 4 coronaviruses that are cause of the common cold. And then we use that structure for protein engineering to stabilize or identify where to put the stabilizing substitutions. That directly came from work from Johannes Langendijk at Janssen, where for the respiratory syncytial virus F protein, they also put a proline in this similar region between the central helix and the first element of the heptad repeat. And that was a great substitution for RSVF, one of the best ones. It boosts expression like 10 to 20-fold, helps keep it in the pre-fusion state. That work was also inspired by John Moore and Rutger Sanders, who put a similar proline in GP41 of the HIV envelope. Those are called the SOCIP trimers, and the IP is the isoleucine to proline. And then that, even before that, is Judith White's work on influenza hemagglutinin, where she also put 2 prolines in sort of a similar region of HA2.
So you stand on the shoulders of giants.
Always. There's always cool research to go learn from.
So have you tried a bunch of different other substitutions as well, or is at least the initial focus mostly there?
We did try many other types of substitutions that failed. And in part, it's because we had the structure of HKU1, but we're performing all the protein engineering in the MERS spike. And those only have around 35% sequence identity. So a lot of things we were trying, like disulfide bonds, you have to be really precise. You have to be within an angstrom or 2 in order to get them to form. And those just weren't forming. But prolines only require conservation of secondary structure. And so that region is highly conserved in secondary structure. The central helix, there's a coil region, 2 non-conserved amino acids, and then this other helix. And the 2 non-conserved amino acids are where we put the prolines.
So you said there's only 35% amino acid homology between HKU1 and MERS, was it?
Yes.
How about the structural homology? Are they much more similar? I don't know if you can put a number on that.
Yeah, I can't remember exactly like what the root mean square deviation is. They overall look similar as you would expect.
Right.
If he told you the root mean squared, would you know what it means?
That wouldn't mean a thing to me.
I didn't think so.
I love hearing him talk about this because it's like I can see in his head he's probably walking around inside the structure, right? You live and breathe.
We've solved a lot of spike structure.
I think he's thinking about lunch probably.
Especially if the daylight savings.
You said you get higher expression. So what does that mean? Is it just more stable? You have less degradation, or do you actually synthesize more protein?
Yeah, we haven't looked specifically at that. So our readout in that case is just the amount of protein we purify from a given volume of cells. And so in that context, either getting more protein produced per mRNA or having less of it degrade would result in a final higher yield.
What are you producing it in?
We usually either use Chinese hamster ovary cells or HEC293 cells.
Okay, so I'm thinking that if it folds differently, the cells might get rid of it or something like that. Does that make sense?
They do have protein quality control, and so generally only well-folded protein makes it through the ER. Golgi gets secreted. Yeah.
So how does the 2 prolines work? What do they do structurally?
Structurally, it's a region that exists as sort of a flexible coil. Maybe think of it more of like a spaghetti noodle. And then it has to convert, that region has to refold into an alpha helix. And so you need some flexibility in there. And proline is the most rigid of the amino acids. So we think that just makes it much more difficult for it to refold. And prolines are often known as helix breakers. They're not found in helices. So maybe it just increases the activation energy for it to refold into a helix.
So when you're talking about refolding, you're talking about refolding from the pre-fusion to the post-fusion conformation?
Yes,
is that right?
yeah. Pre-fusion or even things like pre-hairpin intermediate, like things
Right.
along the
And
way.
it's that pre-fusion conformation that you want to stabilize to make a good antigen.
Yeah,
for
that's
a
what's on
vaccine.
the surface of the virus. That's the active form. That's what's going to bind receptor ACE2. And
So if you immunize animals with proline and without, you get better immune responses or more relevant immune responses with the prolines?
we obtained higher neutralizing antibody titers than without. And we showed that for MERS back in our 2017 PNAS paper. And then people have seen it to different extents. We also have a 6 proline version as well for SARS-CoV-2 that elicits even higher neutralizing antibody titers. Johnson & Johnson, they tested with Dan Baruch in non-human primates different spike variants at the beginning of the pandemic and showed that the 2 proline one was better than wild type. And so that was
Better at inducing
neutralizing antibody responses, as well as some of the other things they were looking at.
I've always wondered if we had used spike without prolines at all for our vaccines, what would have happened?
We still would be okay. It wasn't. It's not the difference between having the vaccine or not. It's just maybe a more efficacious vaccine. You're allowed to use a lower dose, things like that.
Yeah, okay. Can you back up a bit and talk about what happens when spike binds ACE2, the receptor? Well, it's a trimer, and there's up and downs and conformations. Tell us all that
Okay.
without a blackboard.
Yeah, no problem. So each protemer has a receptor binding domain. And so the trimer has 3 RBDs, and they interact with each other at the apex of the molecule. We know that each of them can go from a down conformation, where the receptor cannot bind because it's sterically occluded, and then it transitions into an up conformation, and now the receptor can bind. So at the basal state, there's some equilibrium of down and up. We think, at least for some of the ones we've characterized, generally the most favorable has 1 RBD up, 2 down. So one molecule of ACE2 binds to the RBD that's up, and now the RBD can't go back down, so it's locked up. And then the second one can flicker up and down, and then when it flickers up, another ACE2 molecule binds. And then we think that happens a 3rd time, so it's sort of like a ratchet until all 3 RBDs are in the up conformation. At that point, the RBDs are no longer interacting with each other, and the S1 subunit is now unstable and falls off of S2. And S2 is this sort of spring-loaded fusion machinery that then begins to refold, elongate, and then it shoots part of its self into the target cell membrane. And now you have the S2 subunits spanning both the viral membrane and host cell membrane, and what we call a pre-hairpin intermediate. And then that begins to collapse in half, bringing the N and C termini of S2 together, bringing the viral membrane and host cell membrane together, causing the membranes to fuse. And at that point, the contents of the virus are now in the cell, and the cell is infected.
So let me get this straight. Is it the RBDs, all 3 of them being up, that really initiates this big conformational change?
Yes, that's the model.
Okay.
I mean, it could be.
Because I was thinking,
even,
as you were describing this, I was thinking, why bother with this up-down stuff? Just be up all the time, okay? But you don't want that because you don't want it to fire off prematurely.
That's right. Yeah, all of these fusion proteins, particularly like the class 1 viral fusion proteins that Spike is a member of, they want to have spatiotemporal control of the conformational change. So they want to make sure it occurs at the right place and the right time. Right. If it triggers prematurely, then you get a bunch of inactive post-fusion spikes on the surface of the virus. And so there's different ways of the virus kind of detecting that it's near a cell. And one way is just to couple the conformational change to receptor binding. So then it knows, I'm here, we're at the cell.
Yeah.
Another way that influenza uses is low pH. So in that case, the virus gets taken up into endosomes. It starts to acidify, and then the low pH causes the conformational change to occur.
So the prolines do not interfere with the up-down of the RBD, right?
No, there's been some data. I mean, maybe for SARS-CoV-2, it slightly increased the proportion that was up, but it's not supposed to.
So at what point in this narrative would
Yeah.
the two prolines affect it?
Two prolines somewhere before S1 shedding. It's a good question. At one point, in the absence of it, if we work with just wild type spike that we purify, that will trigger over time from pre-fusion to post-fusion in solution. But if we have the two prolines, we don't see that. We see just essentially all pre-fusion. So it's helping, I guess, even prevent the shedding.
Got it. So if what did I forgot my question now. If the
You can't be president.
yes, you're right. I will never be
I'm paying close attention to this.
president of anything. So basically, the S1 remains attached to S2 in the presence of the two prolines, right?
Yes, that's what we observe.
All right. Now, yeah. So this is the other question. You say the S1 falls away. So why doesn't the virus particle fall away? Is it so quick, the fusion transformation? Because if S1 falls away, that's bound to the receptor, and then the rest of the virus is S2 that's no longer attached to anything, is it?
But it's already in proximity.
It's very close?
It's already close enough. And presumably there's other attachment, right? There's multiple spikes binding to multiple receptors.
Okay.
And so not all of them are going to trigger simultaneously.
It's interesting. You say presumably, so we don't know, but we guess there's more than one attachment happening, right?
Yes, yeah. And then the N-terminal domain can also interact with different cell surface factors. For some coronaviruses, it's glycans that are on the surface. So you sort of get some weak attachment from the NTD and then more specific protein receptors to the RBD.
But you only need one fusion peptide exposed for it to fuse, right? You don't have to have more than one. Or do we not know that?
That's not something that's been studied. The model is all three fusion peptides from one spike. And then there's still other questions like how many spikes do you need per fusion pore, which is still an open question.
So in terms of neutralizing epitopes, most of them are on RBD, is that correct?
A lot of them, many of the best ones, most neutralization sensitive are on the RBD. There's also neutralizing epitopes on the NTD as well as the S2 subunit.
So the effect of the prolines is not simply to keep the RBD exposed, right? Because it's always exposed.
Correct.
It's just to get more antigen.
More antigen produced and to keep it in the prefusion state to help prevent S1 from dissociating.
You see, you thought we couldn't talk about two prolines anymore, right?
Oh, yeah, we can talk about it forever. And he thought he could only talk for an hour.
Yeah,
He's in big trouble.
We're working on it,
Right,
yeah. So I had another two proline question. Yeah, I can't be president. All right, so it's only in terms of the amount of antigen, because I always had the impression that it's a matter of exposure of the RBD epitopes, but that's not the case. Right?
That's not the case. Right. And we also think there's other things like getting more antigen into the germinal centers.
So many neutralizing antibodies are directed against RBD. So presumably those inhibit by blocking attachment to begin with, right?
Yeah, sort of two different ways, or that aren't necessarily mutually exclusive. Some directly compete with ACE2 binding, and so that's nice because now you prevent attachment. Others can actually bind in such a way that it's mimicking ACE2. It traps the RBDs up and can cause premature triggering and dissociation of S1.
Okay.
But in order to have an antibody doesn't necessarily have to be neutralizing to be good for me, right?
That's correct. Yep.
Okay.
Yeah, neutralize.
This to me gets lost because we're constantly measuring the effectiveness of something in terms of neutralization, but that's only part of the story, right?
Yes, and neutralization is really, it's more of an in vitro assay.
Right.
It needs to be done, well, different labs do it differently. It depends on the cell line you're using, the amount of receptors expressed in those cell lines, the amount of protease, incubation time, other things. So it's just an in vitro metric, but it's easy to do in high throughput. The vaccine efficacy correlates relatively well with elicited neutralizing antibody titers, but ultimately we care about protection. And so there are some non-neutralizing antibodies that are protective, in which case you'll usually need the FC region of the antibody.
Right.
It's easy to measure neutralizing antibodies.
right.
The others are harder to assay, including T
Well,
cells.
yeah, it's
too.
something you can do. It's,
Yeah, that's right. So have you used this 2-proline manipulation for other viral vaccines?
We not necessarily 2 prolines, but we use proline scanning for many other of our stabilization efforts. So I mentioned RSVF with Johannes Langendijk at Janssen. We with Jose Malero used it for human metapneumovirus. Peter Kwong's group have used it for Nipah virus. There's a lot. It's now one of the standard design strategies for stabilizing class 1 fusion proteins is identifying regions, generally capping helices or inflexible loops, and scanning with prolines in that region.
So you put the prolines in different places and you measure the protein production and antigenicity to see which is the best place.
Yep.
So
Yeah, we're generally trying to avoid surface exposed residues. We're trying not to change the antigenicity and reactivity with an antibody.
yeah. So would this be useful for influenza virus HA? So those vaccines aren't great,
It should,
right?
but well, flu HA at neutral pH is quite stable, right? Because its triggering is
It's the low pH,
yeah.
Okay, so it's only for neutral pH.
Yeah, although I don't know all the engineering. There has been a lot of engineering in the flu field, particularly making headless HAs,
Right.
where it's just HA2, and I think prolines have been used there as well.
Okay. So this brings up the whole concept of vaccine engineering, right? Yes. And besides prolines, you can do other things, right? So you've done a bunch of them. Tell us about
Yeah,
those.
we sort of have a standard toolbox of different strategies. So proline substitutions to cap helices and to be in relatively unstructured regions is one strategy. Engineering disulfide bonds. So that way we covalently link 2 regions that would normally be flexible or separate from each other during the pre-fusion to post-fusion transition. Cavity filling substitution works really well. So normally, well-folded proteins don't have a lot of internal cavities or solvent-exposed cavities that makes them unstable. But these molecules have to be somewhat unstable in order to be able to undergo the conformational change. So you can find these little pockets and then fill them with like smaller, like a serine to phenylalanine or to an isoleucine. So cavity filling is common. And then we'll also try to introduce in salt bridges. So maybe there's a free negatively charged glutamate nearby, and then we change an adjacent residue to a positively charged lysine. And now we form a fairly stable salt bridge between them.
So these are all approaches to stabilize the proteins.
Yes.
Interesting. So this is relatively recent development, right?
I'd say over the last decade.
Yeah.
Where there's various servers you can run. We're developing in-house tools as well. The advance of AI/ML has come up with alternative ways of identifying regions of instability.
Okay.
And what I really like is it's rational design, right? You're looking at a structure, and based on the structure, tuning in on places where you can tweak the structure to get the result.
That's right, yeah. We've traditionally used experimental structures that have been determined by X-ray or cryo-EM. We're trying to do more using like AlphaFold models. I think the goal for the future for like pandemic preparedness is to go from a genome sequence of a pathogen to an AlphaFold model of the glycoproteins to in silico protein engineering to design the stabilizing substitutions directly to mRNA, encoding those for a vaccine, and just skipping all the lab work.
Okay, so this you've probably told me you've probably told TWiV and this story many times, but I forget. I can't be president
all right.
either. So
Can we-
I want to hear the story. When the pandemic hit, you've got years of experience with other coronaviruses and other viruses with similar fusion proteins, sort of targeted towards this sort of thing. How can we make a good antigen? Okay. When you discovered that this pandemic was a coronavirus, was that a surprise? What were you thinking? What was your reaction?
Yeah. Well, when we found out, it wasn't even a pandemic, right? It was very early. So, I mean, that was the end of December 2019 when there was reports of these pneumonia clusters in China of unknown cause. And so Twitter virologists were already sort of thinking the two most common would be influenza or a coronavirus. And so I think it was likely it was going to be one of those two. And then we found out in early January when Chinese scientists had actually isolated and sequenced that it was a beta coronavirus that was similar to the SARS from 2002, 2003.
So in terms of primary sequence, how similar is SARS-CoV-2 to, say, MERS or SARS or the other coronaviruses that you had worked on before?
The spike protein sequence identity to SARS is, I think, around 70%.
Okay.
And to the other beta CoVs, it would be more in the 30s.
Okay. So where I'm going with this is that, if I understand it correctly, your major contribution was to take the sequence, okay, and very quickly determine a structure for this thing. And the dogma in the press anyway is, oh, mRNA vaccines are cool because all you need is a sequence and you can make the protein and you got a vaccine. But the structure determination, what's the motivation for that? I mean, is to make sure that the stabilization that you designed into it and the structure really does do what, make a structure, make an antigen that should work?
Yeah, that's part of it. There's sort of several reasons to do the structure.
Okay.
One was that we did want to show that the introduction of the prolines led to a well-folded pre-fusion spike protein.
And you need the structure to make sure of that, right?
Yeah. I mean, especially at that time, there weren't. Sometimes you can get by having a nice antibody repertoire that you can antigenically characterize the protein, but that early in the pandemic, there's only a few antibodies known that cross-reacted, and they were just against the RBD.
Okay.
So it wouldn't really tell you anything about how well the quaternary structure of the trimeric spike existed. So that was important. We were able to also show, I think with some negative stain EM, that ACE2 bound. Other groups had shown functionally, but we were at least able to show an image of ACE2 bound. And then the coordinates were really useful for many people in the field that were designing different molecules for binders, that were trying to understand spike substitutions. Like you want to have, especially as variants started to emerge, you could now map where these substitutions, where the antibody escape mutations were on the structure. So it was important to get that out there.
Okay, great.
But when you saw the sequence, did you immediately know where to put the two prolines?
Yes, because our work before, just based on the sequence alignment, we know exactly where to put the prolines.
So you put them there and that was it?
That was it.
You didn't have to fool around at all? Okay.
Nope.
So you published papers on other viruses. So we talked about respiratory syncytial spike. Is that a two proline?
There's several different flavors of the pre-fusion form of that. So the one I helped design back when I was a postdoc in Peter Kwong's lab in 2013, we call that DSCAV1. So that has a disulfide bond and two cavity filling substitutions. So ours does not have a proline. Janssen's did. They're one of the other pre-fusion molecules. And then Pfizer also has a pre-fusion F as well that does not have a proline.
And these are licensed, some of these, right?
Some of these are.
Yeah, okay. And then Hendra, what did you do to the Hendra spike?
Even Hendra, that was an effort led really by NIH, Barney Graham, and Peter Kwong helped design one of the disulfide bonds in the Nipah virus. And then we've just been looking to try to extend and identify additional substitutions that would work well for the family of viruses. So we do want to make a very good Nipah virus vaccine and Hendra vaccine, but we're always concerned more about pandemic preparedness and the emergence of a Nipah or Hendra-like one in the future. So trying to identify substitutions that work well in all of the family members is a high priority, rather than knowing which ones only work well for Nipah. It's great for Nipah vaccines, but doesn't, in a pandemic response, we wanted to know that the prolines work. And so we were able to identify at least one of the disulfide bonds in the Nipah virus works well in Hendra, I think, as well as Langia.
So Langia is a related virus, right?
Langia is a related virus that emerged more recently, I think just a couple of years ago in China.
Okay, right. I think I remember
They're
that.
they're always emerging.
Okay. I also want to talk about a more recent paper where the title is Prefusion Stabilized SARS-CoV-2 S2 only provides protection against SARS-CoV-2 challenge. So why S2 alone? That's the fusion. That's the part that remains and goes into the fusion state, right?
Yeah, so that's the metastable portion. It's the fusion machinery. Looks very similar to the RSV-F protein, as David Wiesler showed, and they're all the class 1s are related to some common ancestral fusogen, and then they've sort of evolved. So this work is really trying to assess how best, or is it possible to make a pan-coronavirus vaccine, or a pan-beta coronavirus vaccine, or a pan-SARS-CoV-2 and all the variants vaccines. And there's different ways of doing that. You can multiplex, you could immunize with like 6 different spike proteins, or RBD nanoparticles containing a bunch of different RBDs. So that's one way to try to get breadth, is just show your immune system many different flavors. Another approach is to try to focus the immune response on the region that's most conserved among all the different spike proteins, and that's S2. So S2 is fairly well conserved, and this is something that has been tested for in the influenza field. So as I mentioned, groups have been making these headless spikes to try to generate antibodies against the stalk. And some of these antibodies are incredibly broad, neutralize many different influenzas. And we thought, well, let's try something similar. We need to pop off the top of S1 and identify substitutions that help retain S2 in the pre-fusion state without the S1 belt that kind of sits on top of it. So really like to think of S1 as this fusion suppressive cap, like a mushroom cap, sitting on the spring-loaded machinery. And so we thought it would be some fun protein engineering to try to make a molecule where that S1 belt doesn't exist and S2 doesn't prematurely trigger. So we were able to do that and test it. And it's a nice molecule. It's a nice reagent. We've used it to isolate and characterize many different S2-directed antibodies. In mice, it actually looked pretty good. It was protecting against SARS-CoV-2 challenge, some partial protection against SARS-CoV challenge. We're not getting a lot of neutralizing antibody titers because, as you mentioned, many of them are directed against the RBD, and that's not even there in that antigen.
When you talk about testing this in mice, are you testing it as an mRNA-based immunization, or are you making protein?
That was protein-based, so subunit-based vaccine.
So this brings me to another thing. I was surprised, actually, that the RSV vaccines were protein vaccines. I thought mRNA was going to be the solution to everything, right? Okay, so how do you decide, or what are the considerations as to whether you're going to try and make a vaccine as an mRNA-based vaccine or a protein vaccine?
You're asking me or a company?
I'm asking your opinion.
Yeah. I mean, all the different modalities have strengths and weaknesses, right? And I think maybe that's something that's lost, is that mRNA isn't like the best. It has strengths. Speed is one thing, right? So just the manufacturing is just much faster than making a stable chill cell line and working up the bioreactors and purification procedures, whereas mRNA is mRNA. And so those purifications, multiplexing also works really well. So you can test like 6 mRNAs in one single shot. So those are some of the advantages. But protein-based, adenoviral, those are still very good vaccines, robust immune responses for protein plus adjuvants that contain like TLR agonists and other things. For RSVF, that work we designed the antigen back in 2013, right? And that was in the early days of mRNA vaccines, and so that's just been going through the normal decade-long vaccine development process. The GMP manufacturing, the stable that took a couple of years, and then phase 1, phase 2, phase 3. So both the GSK and Pfizer RSV vaccines are purified protein subunit. But Moderna has an mRNA-based one. It's a second-gen version of the molecule we designed. I think that's likely to get approved later this year. So there will be an mRNA vaccine, I would think, for RSV.
What has been the challenge with RSV? This has been going on for a long time. Have there been vaccines that didn't work well, right? I mean, what makes the difference between, in that particular case, one that works and one doesn't?
Yeah. There's a long history of RSV vaccine development, dating back to when it was isolated back in like 1955, 56, and one of the first vaccines to be tested was a formalin inactivated RSV vaccine, and that was sort of common for the day and worked for some viruses. And so in that case, you just grow up virus, lots of it, and then you treat it with a chemical called formalin, usually at an elevated temperature, and you let it sit for a couple of days, and then that inactivates it. And then they tested that, and when they tested it in infants who had not yet been infected with RSV, and ended up priming for an exacerbated disease when they were naturally exposed to RSV the following season.
Right.
So the vaccine itself initially was well tolerated, your normal symptoms, but it wasn't until they were just naturally exposed to RSV that the disease they got was much worse than the control group. So I think 80% of the infants who received that vaccine had to be hospitalized, compared to only 5% in the control group, and 2 of the infants died. And so that there's lots of studies as to why that occurred. It was both the B cell responses and T cell responses were suboptimal, and you're getting more of like an allergic type response. The vaccine did not elicit high titers of neutralizing antibodies. It elicited a lot of antibodies, but these just bound and didn't neutralize. Barney Graham's group showed that that process of heating it in the presence of formalin for 2 days basically converts all of the F protein from pre-fusion to post-fusion. And so you're just losing a lot of the really neutralization sensitive epitopes. And so rightly so, that's really put a lot of scrutiny on RSV vaccine development, especially for that very young, the seronegative infants. And so these RSV vaccines we have now have been approved for the elderly and for pregnant women, but we still don't have one approved for seronegative infants.
People are being real careful.
Yeah, they got scared.
Yeah.
I mean, based
Yeah,
on this,
that's
right?
scary.
Well, they should. Yeah.
I mean, Barney once told me that
Yeah.
people it took 50 years to get over this.
Yeah.
Right. That's what a
So,
bad.
I mean, some people may come away from this whole pandemic experience thinking, oh, it's easy. You just identify the pathogen, make this mRNA vaccine, and we're good, right? No, it's a lot more complicated.
It's a lot more complicated. And some of the viruses that we still don't have vaccines for, there's reasons why
Right.
mRNA is not a silver bullet for them. There's just fundamental issues with the immune system and the virus, or different aspects of the virus.
There's also a perception problem, right? And about 30% of the population is afraid of mRNA vaccines, and then certain people make them more afraid by publishing stuff that isn't correct, right? Like it's going to integrate in all this stuff. So I mean, we got this on TWiV all the time. They want protein-based vaccines. So I think we need to have both if we want to cover everyone.
Yeah, but I think options are good, right?
Yeah, options.
Obviously, we want efficacious vaccines that people take. So it's not vaccines that save lives, it's vaccinations. So having vaccines in vials that aren't in people's arms don't help. So I think whatever we can do to make vaccines people will take,
Yeah. So getting back to S2, so the antibodies that do neutralize, you said they're not very good at eliciting neutralizing.
The prefusion S2 antigen did not elicit particularly high neutralizing antibody titers.
But there were some newts, and so how do they work? Do they block fusion?
Yeah, they interfere with the fusion process. So they'll bind and either prevent the conformational change to the pre-hairpin intermediate, or they prevent the collapse of the pre-hairpin intermediate to the post-fusion state. Something like RSVF doesn't even have a fusion suppressive cap. Like it just is essentially S2.
Right,
and so those antibodies just bind and hold on to it. And now it can't trigger, it can't adopt the pre-hairpin.
I'm imagining that some of these antibodies made to just a stabilized S2 structure may not even have an opportunity to interact with the native protein. Is that right? I'm thinking of S1 actually occluding the binding of some of these antibodies. Or
yeah, that's right. Yes, so the ones that have been shown to be neutralizing, they generally aren't interfered with by S1. They're binding a region near the fusion peptide or a part the stem helix region, so it's kind of far away from S1. It does appear that you need some breathing of these regions to sort of dissociate transiently and have the antibody bind. We and others have isolated some antibodies that bind the apex of S2 that is normally covered by S1. And those are generally non-neutralizing because they just don't have access to it. But we've isolated them from people. So humans are being exposed to pre-fusion S2 in sufficient amounts or time to actually generate antibodies against this.
so using what you know about what makes a neutralizing antibody against S2, can you now design an antigen that's even a tighter target, if you see what I mean?
Yeah, I think so. It might have to be more focused on individual epitopes. And I have seen some papers on that. So we've been trying to engineer the whole immunogen and just kind of get antibodies everywhere. You can also make more of an epitope-focused one, where you make an antigen that's just the stem helix or just a part of the fusion peptide. That can work. Bill Schief and others, Bruno Correa, have shown that for HIV and RSV. The problem is that you are creating an immunogen where your entire vaccine response is against one
Right,
epitope.
I'm thinking you might outsmart yourself. Right?
It could be very easy to evolve away from that.
Yeah.
That's why I wanted to ask you about these S2 epitopes. Do they change compared to S1?
Much slower. There are some changes, like Omicron has a couple of changes. Sometimes that's more to do with regulating the free energy, the change that is actually escaping from an antibody. So some of these might be compensatory changes to make up for a change somewhere else that was needed to escape from an antibody.
So if you can, as you said, humans make antibodies to S2.
Yep.
So presumably you can pull those out.
Oh yeah. And people have pulled out some like very broad neutralizing. They're generally not as potently neutralizing as the ones against the RBD, right? Because the ones against the RBD are preventing the very first step, generally attachment and the triggering process. The ones against S2, you're already going to have the virus stuck to the cell. Right. ACE2 is going to be there, and so the antibody is going to bind to S2. If it falls off or it doesn't bind during that window, then it's going to fuse, go to completion.
So you think that S2 vaccines are not going to be useful for broadly protection against multiple beta coronaviruses?
I think it's still early.
Yeah.
I think it's still early. We have not seen a benefit to immunizing with S2 only. So in our study, we also included our 6-proline stabilized version of the spike protein, and that did as well or better. And in part because that also has S2, right? Like prefusion S2 is there.
Yeah.
So you're getting those plus some of the neutralizing NTD-RBD antibodies.
Are there other approaches besides S2 that could give? You mentioned multiplexed RBDs, right?
Yeah. So that's an approach people are trying with these self-assembling nanocages.
Yeah.
They have many different copies of RBDs sticking out. So Pamela Bjorkman at Caltech has published on those. And so that's potentially one way of doing that.
Is there anything else that you can think of?
I think I'm going to admit some things and make people mad, right? So I think Ralph Barrick's group was doing some work on chimeric spike proteins, where you could take the N-terminal domain from MERS and stick it to a spike, the rest of S1 from SARS, and then have S2 from HKU1 or something. So try and
Chimeric molecules.
Yeah,
Okay.
chimeric molecules, maybe some T-cell based approaches.
I was just going to bring up T cells because this whole conversation we've been talking about antibodies.
This is going to be a short T-cell
And there's a-
conversation.
It's a B-cell guy, right? He's all about antibody epitopes.
But T cells are absolutely important.
Yeah.
Yeah.
I want to talk a little bit about the variation that we saw. So in the beginning, there wasn't much antigenic variation, but as more and more people got immunized or infected, suddenly there was a lot. Was that surprising to you? Did you go, oh my gosh, I didn't realize this could happen? Or did you say, I knew it would happen? Or somewhere in between?
Maybe somewhere in between. Like we would expect some amount of drift, especially as it's replicating kind of unchecked globally, and then you're putting some immune pressure on it. And certainly you see that with flu, right? That's one of the reasons we need to update the vaccines every year. So it's also an RNA virus. These things can change.
So do you think we're going to have to update SARS-CoV-2 vaccines every year as a consequence?
It's a good question. I think some of the papers are coming out soon or in preprint, looking at the immune response now. And it is a fairly heterogeneous population, which is interesting, right? Because you have some people that have been infected 1 to 4 times.
Yeah, sure.
And then with or without 1 to 6 vaccines. And so what do you recommend to those people versus a newborn who hasn't been exposed to either yet? We have seen quite a few new viruses kind of explode on the scene, JN1, BA286, with lots of changes. And they've replicated and kind of taken over the circulating strains, but we haven't really seen large increases in hospitalizations. So I feel like we have a pretty robust immune response now, particularly if you have the hybrid immunity, if you've been infected, if you've been vaccinated and boosted.
So it's possible we might see less antigenic variation going forward. You think that we haven't exhausted all positions?
No.
Right?
Yeah, and I think we'll continue to see evolution. I don't think that's going to stop. It's just a matter of like, what is the frequency with which we need to reformulate the vaccines to include a new circulating strain?
Yeah, I know it's hard to predict, but it's just interesting to talk about. I mean, a change like Omicron probably isn't going to happen all that often, I would guess, but who knows?
Who knows?
Right.
I don't know. I see some tree and there's an outlier they find in like wastewater surveillance.
So if you look at the structures of these variants from the first day to the present, is there anything fundamentally different about how they hit ACE2 and how they undergo fusion?
No,
Nothing.
that has to be conserved. Yeah. So these are just more generally surface changes, just to reduce antibody binding, maybe improve cleavage, efficiency, things like that. But the overall function has to be the same.
Now, early in the pandemic, relatively early, a number of monoclonals were made, therapeutic and even prophylactic monoclonals, right? And then we stopped doing that because of the variants, right? And nobody seems to be doing that any longer. Well, why not?
I think it just got expensive. Again, especially if you're doing the protein-based antibody therapies where you need to make stable cell lines, work out purification procedures. It's a lot of lead time and testing only to then be obsolete by a new variant emerging.
Can you make monoclonals against conserved residues, though?
Yeah, and that's a big push now, and I think it's always tough, right? So yeah, it has been just continuously people publishing, here's an antibody that works against all known variants, except the next variant where it doesn't work. And then there's a new antibody that works against all those known variants.
Except the next one,
Yeah.
So companies don't want to take that risk, basically.
Yeah. And I don't think the antibodies were particularly is widely used.
Well, certain people, like immunocompromised patients, can't be immunized, so it's good for them, right?
Oh yeah, there's absolutely need cases for them.
And then if you can extend the life, you could go 3 months, then that's really nice that
Yeah.
you can do that, right? But what about, you talk about the difficulty in production. You can make single chains in bacteria, right? So that should be cheaper. Is anyone, to your knowledge, looking at that?
I'm sure people are looking at that,
Okay.
but
You
at
don't
least
know.
the ones that we've been discussing that were approved from Regeneron and Lilly and others, those are all normal IgGs, in part because you do want that FC effector function.
Okay.
You do want the half-life extension.
Got it. Do you have any other
I'm
COVID?
good.
I have one.
I have to say, I actually think about this stuff when even outside of this sort of context, and this has been great because I've had a whole bunch of questions in my head that have
Got them
been
answered.
addressed. Yeah.
What else are you interested these days? Only coronaviruses?
No. We've been winding down coronavirus
Yeah.
research for we're just getting out the couple final papers now. So, what are we working on? I think really trying to push the direction of structure-based vaccine design and to see how far we can take it to what it can be applied to, right? Like every time there's sort of a new technology, you get a burst of new vaccines being developed, whether that's glycoconjugates, restriction enzyme, molecular cloning, and things. So it's kind of so we're trying to do more challenging things. So we've done a lot. All the things we've talked about so far are class 1 viral fusion proteins. So we've been doing work on class 3 viral fusion proteins, like the GB proteins of herpes viruses. And so we have a preprint out there. At least based on our initial results, our pre-fusion molecule did not elicit better neutralizing antibody titers than post-fusion. That's just an N of 1. We need additional people are following up. We'll be following up with other things, so we'll see if maybe that doesn't hold true for all class 3s or just the subset of class 3s we were looking at. We're also looking at various class 2 viral fusion proteins that form higher order assemblies, which are kind of interesting. So we've been doing work on bunioviruses, Crimean Congo hemorrhagic fever virus, and others, and these tend to form tetramers or pentamers, and so trying to figure out, can we actually make soluble versions of these whole higher order assemblies of the fusion proteins. And then we've done some work on pertussis, trying to think, can we start applying structure-based vaccine design to bacterial pathogens with the rise of antibiotic resistance, trying to figure out what are the surface proteins or toxins, where do antibodies that humans make bind, can we engineer those to express better, be more stable? Also, is there engineering opportunities for trying to encode bacterial proteins in mRNA for mRNA vaccines? A lot of those proteins need to be folded in bacteria, secreted through secretion systems that our cells don't have. And so those are some of the different things we've been looking at.
I want to talk for just a second about AlphaFold because I have, no experience with this. And for audience out there in the cloud, these are, is it fair to say these are computer programs that take a primary amino acid sequence and predict what the three-dimensional structure would be, right?
That's correct.
Which has been a holy grail
That's
forever.
That's the structure prediction.
Yeah.
It helps solve structure prediction.
If that were perfect, it would obviate, as I think you said early on in this, all the lab work necessary to determine structures.
Kind of. It obviates some of it, maybe.
So this is my question,
Yeah.
is how does this now play into what you do? What's the balance between, how good is AlphaFold? What's the balance between its utility and your actual laboratory structure-based work? How do you use it?
Yeah. It's an incredible tool we use a lot. Sometimes you don't know how good it is until you experimentally solve the structure and then see where it differs. Strengths include really smaller proteins that are a single domain. It really just nails those. It has a harder time on something like a fusion protein that exists in 2 states. Right? It's a bit unfair. You're giving it an amino acid sequence and saying predict the structure, but we know it adopts at least pre-fusion or post-fusion. Sometimes then it returns kind of a hybrid where some parts have moved more like pre or post. Higher order complexes it has more difficulty. But I mean, these are all active areas of investigation where people are trying to push it, right, to form. There's alpha-fold 2 multimer. There's things where you can include mass spec cross-link data to help improve the contacts of which amino acids should be which others. There's been advances with small molecules, DNA. I think Rosetta-fold all atom was just released from the Baker Group, which can help now take into account small molecules and nucleic acids, which is really exciting. But a lot of times, dynamics, the structural biology is really helpful because you might be able, especially when you're working with machines, enzymes, things, you might be able to trap different states experimentally. And AlphaFold's still just going to give you a structure, or maybe some ensemble, but it's hard to put in order. And so you might be seeing more of the experimental going at these like conformations and ensembles.
That's really exciting. It's so cool.
It also just helps actually making a lot of our structures better determined. So even when we experimentally determine a structure and we get a map of the electron density or Coulomb potential, we used to just have to maybe take another related structure or build into that de novo. And maybe some groups are better than others. There'd be misbuilds and things. Now I think many groups just take an AlphaFold model and stick that into the map and then tweak and modify and adjust. And I think that's just been resulting in much higher quality structures.
So when you get a structure, can you go and tell AlphaFold, hey, dude, you got this wrong, okay?
No, I don't think there's feedback, at least not from me.
Yeah, it's not chat AlphaFold.
Right.
Yeah. There have been some iterations and things. We're trying to use AlphaFold plus the map is a constraint and try to like iterate through that. Shore Shears' group also released something called Model Angelo, which uses AIML. You just feed it the map, and it starts building the structure into it. And so these are all just great tools. It allows us to focus less on that part, and generally the structures are of higher accuracy, particularly for groups or people that have less experience doing it. We're just getting more accurate structure.
Did you say Model Angelo?
Yeah.
That's great.
It's good.
Michelangelo. What's the fastest you can solve a structure by cryo-EM these days?
The fastest. Starting from when?
If I give you protein.
Right
protein.
now.
Oh.
Protein.
I mean, you can have it by tomorrow.
Really?
It's going to depend on a lot of, but I mean, you said the fastest, right?
But
so
the
I'm
protein
assuming
production is hard, right?
Yeah,
Can be.
we're really sample limited, right? So if you give me a good, well-behaving biological specimen that doesn't require too much optimization of freezing, we'll get nice images. We'll start getting like on the fly data processing is starting to generate 3D volumes, and then with some of these tools, we're auto building. So a lot of it really is down to like sample prep. Wow. Like, can you produce the sample in the purity that you need, in the conformational homogeneity, without parts of the complex falling off or dissociating or having it fall apart on the grid?
Also, going back to the herpes virus, I had one question. Why is Shingrix such a good vaccine? That's a spike protein, right? A single spike.
No, it's the GD protein.
GD.
Yeah, it's interesting. It seems to focus a lot less on neutralizing antibodies and more on protective antibodies of FC effector functions.
Which was just by chance that it turned out that way, right?
I don't know the history of the development.
But structurally, it's no different than any other viral membrane glycoprotein. There's nothing unique about it.
No.
Okay.
I mean, it is
because it's really good, right? It has really high
it's a fantastic vaccine. Yeah.
All right, I have one more question. What would you have done if you hadn't been a scientist?
That's a good question.
You play guitar?
I didn't know. Very few useful hobbies. I don't know. Hopefully, maybe the MD route or something.
No, you don't want to be a doctor.
No.
Well, that's why I'm not.
You have nothing else that you'd really want to do, right? Except
like right now, I think I'm super interested in robotics.
Okay.
So, yeah, because that just involves like coding, robots, electronics, like that. That's a bunch of things I like.
you had a robot at the science mill.
I saw they were doing battle bots at South by Southwest.
Yeah, they do. In fact, there's a big exhibit by the Army.
Okay.
And they have all of their robots that they use for combat. It's really amazing. And they do battles with them and so forth. It's quite interesting. All right. That is a special episode of TWiV at University of Texas, Austin. All right, you can find TWiV at microbe.tv/TWiV. If you want to send questions or comments, TWiV at microbe.tv. And if you enjoy these kinds of science educational programs, we'd love to have your financial support to make more of them, to pay for our expenses. We have some interesting new podcasts coming out. We'd love your help, microbe.tv/contribute. Our guest today from the University of Texas, Austin, Jason McLellan. Thank you for joining us.
It's been my pleasure. I'm looking forward to my jacket next time for the five timers.
Okay, we'll be on that. It's no problem. Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Always a good time and a real privilege to be here. It's really
Thank
great.
you for joining. Thank you for driving us around.
Yeah, sure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.